Previously in the course, we've discussed the importance of exchanging materials between organisms and an environment. And most recently, we've been thinking about how this is done in multicellular organisms, why diffusion alone is not going to be sufficient to supply the needs of the organism above a certain size. So now that we know our way around a plant body, we know how it's organized, we know developmentally how that plant was put together through its life cycle, now we can start to think about these systems of material exchange in plants. So we're going to be talking about resource acquisition and transport in vascular plants in particular. And I'm breaking this up into 3 web lectures. The first one is going to talk about the general principles of material transport in plants with an emphasis on short distance transport. In the next web lecture, we're going to be looking at bulk transport in the xylem. And then finally, in the third one, we'll look at bulk transport in the phloem. So how do we get what we need from the environment? So plants have various adaptations that aid in the acquisition of resources, including water, minerals, carbon dioxide, and light. And the success of plants depends on their ability to gather these resources from their environment and transport them to where they're needed. So if we think in an evolutionary context of the evolution of plants, how this might have come about. So the algal ancestors of land plants absorbed water, minerals, and carbon dioxide directly from the surrounding water, and they needed to be close enough to the surface to be able to also collect light as it filtered down through the water. When we start to move onto land, these non-vascular land plants, the ones without vascular tissue, lived in shallow water and had aerial shoots that poked up out of the water to collect oxygen for respiration and also to be better able to collect photons from sunlight. So natural selection would have favored taller plants with flat appendages, multicellular branching roots, and efficient transport. Why? Because when you start to get dense assemblages of photosynthetic organisms, things living close to each other, then you start to have shading of photosynthetic plants by other plants. And the taller you can get, the higher up above the other plants you can get, the more sunlight you can get, the less you're going to have to worry about being shaded by other plants. An increase in height is going to be a major advantage. Being able to increase the surface area of those leaves is going to be a huge advantage in collecting sunlight. And as you get taller, you need a more elaborate branching root system to hold that plant in place and to supply enough water for that taller, larger plant. So the evolution of the xylem and phloem in vascular land plants made possible the long-distance transport of all of these materials that are being gathered from the soil, the water, and minerals to move up the plant to the leaves where they're needed for photosynthesis and transport of products of photosynthesis, distributing them throughout the plant. So now let's first think about adaptations for light capture. So stems serve as conduits for water and nutrients and also as supporting structure for the leaves. And both the length of the shoot and the branching pattern are going to affect light capture. And so we find that there's a trade-off between growing tall, so you want to get up and above other photosynthetic organisms to get the maximum amount of light, but also the branching means that you're going to have a larger total number of leaves, and that's also going to help you with photosynthesis. And there's a limited amount of energy available, so there's going to be a compromise between allocating resources toward growing tall compared to growing more branches and therefore potentially more leaves. So the more energy invested into branching, the less energy is available for growth in height. Both of these are desirable things in different ways, in different contexts, and there's going to have to be a balance depending on the specific circumstances that a plant finds itself in. So there's generally a positive correlation between water availability and leaf size. So in places where there is a very wet environment, remember that the leaves are the sites of evapotranspiration, so evaporative water loss comes from the leaves. So if you're in a really wet environment, you can afford to lose a lot of water through evaporation and having a larger leaf size is definitely a benefit in terms of gathering sunlight, and so it's fine to have that extra leaf area where you're losing water because there's always more water available. Conversely, if you're in a very dry or a very cold climate, there may be not so much liquid water available to replace the water that's lost, so you need to reduce the size of those leaves to minimize water loss. So the arrangement of leaves on a stem is also really important to the effectiveness of light capture. And we refer to the arrangement of leaves as phyllotaxy. It's the arrangement of leaves on a stem, and it's a species-specific trait important for light capture. So any given species of plant is going to just have one of these different kinds of phyllotaxy. So the different arrangements can involve just having a single leaf from each node. So for this is alternate spiral, we get one leaf from each node. Also, alternate distichous is just one leaf from each node. It's going to come from opposite sides of the plant in an alternating fashion. In alternate spiral, it's going to kind of spiral up the plant and not have leaves directly one on top of each other as you alternate. You can also have arrangements that have two leaves coming from each node, and these are called opposite leaves. So from each node, you're going to have one leaf on either side. And you can also have entire whorls of leaves at each node, and this is called a whorled phyllotaxy. So most angiosperms have this form here, the alternate spiral, where the leaves kind of spiral up. This minimizes the problems of higher leaves shading out lower leaves. And the arrangement of these leaves is usually at an angle around 137.5 degrees between one leaf and the next, if you go around that circle, and likely this minimizes the shading of the lower leaves. So the total area of the leafy portions of all the plants in a community affects the productivity of each plant. So the extent to which different plants are being shaded, the extent to which different plants have a large leaf area that can collect sunlight is going to be a factor in the productivity. If a plant finds itself in a situation where it's not getting enough sunlight, if respiration is using up more sugar than is being produced by photosynthesis, then the plant might respond by shedding off the lower shaded leaves that are not really very photosynthetically active but are still taking up resources for respiration, getting rid of those non-productive parts and just maintaining the parts that are actively photosynthesizing. So light absorption is affected by the leaf area index. So this is the ratio of total upper leaf surface of a plant divided by the surface area of land on which it grows. So this, if you look at the plant from the top and you see something like this, you're seeing lots of ground underneath. This is going to have a lower leaf area index than a plant like this, where when you look down from the top, you see mostly leaf cover. So this is going to absorb a lot of light, very little of the ground underneath it. This is going to have a high leaf area index. But also when you get a high leaf area index, you're also getting a lot of shading of the plant. So leaf area indices higher than 7 result in shading to the point that you start to get self-pruning, because you're going to have a lot of the lower leaves that are just not contributing to generating energy. So the orientation of leaves also affects the extent to which they can capture photons. Remember, you're going to have maximum photon capture when the broad side of the leaf is facing directly toward the sun. You're getting the most direct photon hits in that way. In low light conditions, horizontal leaves that could be oriented directly toward the sun are going to capture more sunlight. There are going to be more photons that are going to hit that horizontal leaf from the sun coming from above. In sunny conditions, where there's a risk that there would be so much light that the cells could be damaged by the amount of light, then vertical leaves are going to sustain less of that damage from the sun and allow the light to reach the lower leaves as well as the upper leaves. They're going to allow more light to come through and keep those lower leaves from being shaded. So here we have horizontally oriented leaves in this plant that are going to absorb most of the sunlight coming in, and then these ones that are more vertical are going to absorb less of the total sunlight. So now let's turn and think about the acquisition of materials from the soil. So the root architecture and water and mineral acquisition. So soil contains resources that are mined by the root system. The root system is going to actively grow in a way that allows it to maximize resource acquisition. So root growth can adjust to local conditions. So for example, when the roots are growing through the soil, if they detect high levels of nitrate, they're going to start branching like crazy. Spread out as many lateral branches as they can to take advantage of those high levels of nitrate in the soil. If they're growing through and they're not detecting high levels of nitrates, they're just going to grow right through it and keep growing down until they can find a better source of resources. And roots are also going to associate with different fungal species. Remember that fungi produce these very, very fine, long filaments called hyphae that spread out and have a huge surface area for absorption. And these associations with hyphae are called mycorrhizae, and these associations are going to help the roots to be able to absorb materials from the soil more effectively through these mutualistic, mutually beneficial associations with these fungi. So mycorrhizal fungi increase the surface area for absorbing water and minerals, and this is especially going to be true for phosphate. And it turns out that when we think about the origin of plants on land, the fossil record shows that this happened in association with the invasion of land by fungal species. So mutualisms with fungi enabled plants to actually make use of the resources in the soil on land. So if we think about short or long distance transport of materials needed by the plant, they're going to be using very different mechanisms. So there are two major transport pathways through plants. One is called the apoplast, and the other is called the symplast. So the apoplast consists of everything external to the plasma membrane. So it includes cell walls, extracellular spaces, and the interior of dead cells, such as vessel elements and tracheids. The symplast consists of the cytosol of all the living cells in a plant, as well as the plasmodesmata. So think of the continuous living part of the inside of the cell, and it's going to include the plasma membrane. So the three transport routes for water and solutes through the tissue of the plant are: one, the apoplastic route through the cell walls and extracellular spaces. So if you follow this yellow line, you can see it's sort of skirting around the cells, staying in that apoplast as it moves through. And in this close-up view, you can see how it goes along through the cell walls, through the extracellular spaces to move around through the plant. The symplastic route is the route in which water and solutes cross a plasma membrane once and then travel through the cytosol of the different cells. And they're going to go from cell to cell through plasmodesmata. So we can see this blue route here that's going inside the cells. You can kind of see the little hole here, the plasmodesmata that it's going through to go into the next cell. And it's going to go from cell to cell through the plasmodesmata. You can see these openings, the plasmodesmata, better in this close-up view, and the blue route is the one that's going through all the cells. Then finally, not pictured here, is the transmembrane route, which is very similar to the symplastic route, but rather than using the plasmodesmata to move from cell to cell, water and solutes repeatedly cross plasma membranes as they pass from cell to cell. So they're not going to make use of these openings, they're just going to cross the plasma membrane. So thinking about short distance transport of solutes into and out of cells across plasma membranes. So remember that plasma membrane permeability controls the short distance movement of substances. We saw this when we were studying the cell membrane, the resistance that it presents to the movement of materials into and out of the cell as a selectively permeable barrier. As we've seen in other kinds of cells, both active and passive transport are going to occur in plants. The membrane potential that's developed in plants. So remember, the membrane potential refers to the difference in electrical charge or voltage between the inside of the cell and the outside of the cell. In plants, this membrane potential, this difference in voltage across the cell membrane, is established through pumping of hydrogen ions by proton pumps. So a similar mechanism for what we saw in cellular respiration and in photosynthesis. But in this case, ATP is going to be used to run these active pumps rather than what we saw in respiration and photosynthesis, where energy released through an electron transport chain was running these proton pumps. These are going to be run by the energy from ATP. So this is in contrast to how that membrane potential is developed in animals, which was, remember, using that sodium-potassium pump, pumping out 3 sodium ions for every 2 potassium ions that are pumped in, creates that difference in charge, negative on the inside, positive on the outside. So remember that these electrochemical gradients are a source of potential energy. They have the potential to do work, to move things around. So plant cells use the potential energy that's stored in these proton gradients and the membrane potential to co-transport other solutes by active transport. So they're going to use this potential energy to move things into the cell. Plant cell membranes also have ion channels that allow only certain ions to pass as needed. So here's a diagram of these different kinds of mechanisms. So here we see a proton pump that's actively pumping protons from the cytoplasm into the extracellular fluid using ATP for every single proton that's being pumped out. Here's an example of using the potential energy stored in this hydrogen ion gradient to co-transport sucrose into the cell. So this is a co-transporter protein that will allow the hydrogen ions to diffuse through along their electrochemical gradient, but only if there's a sucrose also bonded that's going to come along with it. So remember this idea of co-transporters. So hydrogen ion electrochemical gradients can also be used for the co-transport of charged molecules of ions. This is the same mechanism. This channel will allow protons to move along their electrochemical gradient, but only as long as they're accompanied by this needed material that they need to transport into the cell. And then here's an example of an ion channel. Here we have the negatively charged interior of the cell with an accumulation of potassium ions, low concentration of potassium ions outside. They're going to move along their concentration gradient from inside of the cell to outside of the cell. The other major driver of short distance transport across cell membranes is osmosis. So you'll remember osmosis from when we were discussing diffusion earlier in the semester. It's the diffusion of water into or out of a cell that's affected by differences in solute concentration and pressure. So water potential is a quantity that includes the effects of solute concentration and also physical pressure. And water potential is going to tell you whether the water is going to move into or out of the cell, so the direction of movement of the water. So water is always going to flow from regions of higher water potential to regions of lower water potential. Always remember this. We can think of this water potential as potential energy, the potential for doing work. When it has high water potential, it's going to have a higher tendency to move somewhere. When it has a lower water potential, it's going to have a lower tendency to move somewhere. So it's always going to move from higher water potential to lower water potential. So again, potential refers to water's capacity to perform work. So if it has a high capacity to perform work, it's less stable, more likely to move than when it has a lower capacity to perform work. So we use the Greek letter psi to stand for water potential, and it's measured in a unit of pressure called the megapascal. By definition, the water potential equals 0 for pure water with no solutes at sea level and at room temperature. All other water potentials are measured with reference to that default state. So now let's look at these two components that contribute to water potential. So both solute concentration and pressure, physical pressure, affect water potential in plants. And this is expressed by the water potential equation. So the total water potential is going to be equal to the contribution of the solute concentration, so we abbreviate that as psi sub s, plus the contribution to the water potential from physical pressure, and we abbreviate that as psi sub p. The solute potential, psi sub s, of a solution is proportional to its molarity. And we also refer to solute potential as osmotic potential. It's going to contribute to this total water potential by the difference in solute concentration that drives osmosis. Pressure potential is the physical pressure on a solution. So pressure potential can be positive or negative. So a great example of this is using a syringe. You can have suction pressure when the plunger of a syringe is withdrawn, and so the fluid is drawn in under negative pressure, that's suction pressure, when the plunger is pulled up. It's going to be under positive pressure when you push that plunger down and push it out. So you can think of negative as being pulling pressures and positive as being pushing pressures. And then the other kind of pressure that we see in plant cells is something called turgor pressure. So this is the positive pressure exerted by the plasma membrane as it's taking in water and swelling and swelling and swelling. It's going to push against the cell wall, and according to Newton's laws of motion, as long as that cell wall is rigid, it's also going to exert an equal and opposite force back on that plasma membrane, and this is known as turgor pressure. It's a pressure due to the increase in volume of the protoplast, this living part of the cell, including the membrane and the contents. So the increasing volume of the protoplast pushing against the cell wall and the cell wall resisting that pressure. So let's take a look at how to use these ideas. So water potential affects uptake and loss of water by plant cells. We worked on this in more qualitative terms when we were talking about osmosis. Now we're going to look at this more quantitatively in terms of these potentials. So let's imagine we have a flaccid cell. So it's limp, it's not exerting any turgor pressure. And if we place it in an environment with a higher solute concentration, so in this case, in our initial flaccid cell, the turgor pressure, so the pressure potential is going to be 0. The solute pressure due to all the solutes within the cytosol is going to be negative 0.7. So we add those two together, and the total water potential is going to be negative 7 megapascals. So now, if we put this flaccid cell into this environment of a 0.4 molar sucrose solution, so this is going to have a higher solute concentration than the cell had. So in this case, we still have 0 turgor pressure in the solution in which the cell is being bathed, and now we've got a solute potential of negative 0.9. Okay, so remember that water always moves from higher water potential to lower water potential. The cell is less negative than the environment, so it's higher inside the cell. Water is going to move from inside the cell to outside the cell. So you determine the direction of movement by comparing the water potential inside the cell to the water potential outside the cell, determining which one is greater, and that's where water is going to move. So when that plasmalized cell, so when the cell membrane actually shrinks away from the cell wall, remember we refer to this as plasmalized. This is the process is called plasmolysis. Equilibrium will occur when the total water potential outside the cell equals the total water potential inside the cell, and that will happen when enough water has left the cell so that the solute potential inside has reached negative 0.9, equal to the outside of the cell. So plasmolysis, remember, occurs when the protoplast shrinks and pulls away from the cell wall. We say that that cell is plasmolyzed. So now let's take that same flaccid cell with a pressure potential of 0 and a solute potential of negative 0.7, and now let's put it in an environment of pure water. So in this case, the pressure potential is 0, the solute potential is also 0, and so now we've got a total water potential outside the cell of 0, total water potential inside the cell of negative 0.7, so water's going to move from the higher water potential of 0 to the lower water potential of negative 7. Water's going to move into the cell. So now the system will come to equilibrium when the total water potential inside the cell matches the total water potential outside the cell. And so what's going to happen is we're going to develop pressure, turgor pressure, due to the contents of the cell pushing against the cell wall. And so this system is going to reach equilibrium when that pressure potential has an equal magnitude to the negative solute potential. So to equal the zero water potential at the outside. So this will reach equilibrium. There will be no net movement of water at the point when that turgor pressure reaches 0.7 megapascals. So the water potential determines the direction of water movement, but what determines the rate of flow of water movement? This is also going to be an important component to material transfer. And so, although water molecules can and do diffuse across cell membranes, they do so at a relatively slow rate. So what we find in these plant cells are these channel proteins called aquaporins. So these are transport proteins in the cell membrane that facilitate the passage of water, increase the rate. So the more open aquaporins there are in a cell, the faster the rate of water movement, and so the faster these cells will reach their equilibrium state when there's a water potential. So in plants, these aquaporins can be opened and closed using channel gates. And this is going to affect the rate of osmotic water movement across the membrane. So remember, the rate of flow of water across the membrane is going to be proportional to the number of open aquaporins in the cell membrane. So now let's start to think about what we've been calling bulk transport. The book calls it bulk flow. They both mean the same thing, the movement of large volumes of material faster than would be possible using diffusion alone. So efficient long-distance transport across a large body requires bulk flow, the movement of a fluid driven by a pressure gradient. That's what's going to be the driving force for this bulk flow. So water and solutes move together through the tracheids and vessel elements of the xylem and the sieve-tube elements of the phloem. And these branching patterns in the leaves, so all of these little veins you see, all of these little lines, are going to ensure that all cells are within a few cells of the vascular tissue, a distance that can easily be traversed by simple diffusion alone. So bulk flow is enhanced by the structural adaptations of xylem and phloem cells, all of these features we talked about when we were looking at the organization of plant tissues. We thought about these structure-function relationships. So mature tracheids and vessel elements have no cytoplasm. Why? Because the larger the diameter of the lumen of the vessel, the empty space inside the cylinder, the higher the rate of flow can move through that vessel. So cytoplasm, other living materials are just going to take up space on the inside that could be used for rapid flow through those vessels. So the fact that the tracheids and vessel elements in xylem are completely non-living, they need no cytoplasm at all, and that these sieve-tube elements in phloem are alive, but they've lost most of their cytoplasm, and they use these companion cells to do most of the work of the cell and transport materials to them. They're kind of like feeder cells. This is all to increase the effectiveness of that function of transporting materials quickly. And then the perforation plates connect these vessel elements. Porous sieve plates connect sieve-tube elements so that these can move freely through this tube and transport materials long distances. So those are the basic principles of transport of materials in plants. We went into a great deal of detail about the short distance transport. We just touched on some of the basic principles of bulk transport, and now we're ready to go on to look at the specifics of bulk transport, first in the xylem and then in the following web lecture in the phloem.